Hello, so for this video I chose prompt 1 and I'm going to be talking about epistasis. So, epistasis occurs when allele expression at one locus affects the allele expression at another locus. Genes can be epistatic, which means that they mask another gene, and some genes can combine or inhibit other genes to produce certain phenotypes. There are many different types of epistasis, but the one I'm going to be talking about today is called duplicate dominant epistasis, which is where possession of either or both dominant alleles result in having the same phenotype. So for example, having capital A or capital B, or having both capital A and capital B will result in having the same phenotype. To demonstrate, I will enlist the help of my cat Kiki. She's 12 years old, and Kiki works hard and plays hard. Well, actually, Kiki doesn't really work at all, but she does play hard, and sometimes a traditional cat bath is not sufficient enough for her. So in this case, she has two options to get clean. She could either take a bath, or she could take a shower. You might be wondering how this relates to duplicate dominant epistasis. Well, if you think of having a shower or having a bath as alleles, and you think of being dirty or being clean as phenotypes, you could see that either taking a shower or taking a bath, aka possessing either capital A or capital B, will result in the same phenotype, being clean. And if she takes both a shower and a bath, she will also be clean regardless. However, if she doesn't take a bath, she doesn't take a shower, which is, having two lowercase a's and two lowercase b's, she will wind up dirty. So this is the heterozygous cross, and you can see from the Punnett square here that this winds up being in a 9:3 to 3:1 ratio, where 1 out of 16 of the time she will remain dirty because she has both of these lowercase alleles. However, 15 out of 16 of the times, despite having different genotypes, as you can see in the Punnett square, just by having possessing a capital A or capital B allele, she winds up showing the clean phenotype. So it actually ends up being a 15:1 ratio, where 15 out of 16 of the time she ends up getting clean, and 1 out of 16 of the time she ends up getting dirty. This is the second cross, and this cross winds up in a 4:4:4:4 ratio. However, once again, just having a capital allele, either capital A or capital B, or both capital A and capital B, winds up in the clean phenotype. So regardless, 3/4 of the time, Kiki will end up getting clean, but she will remain dirty 1/4 of the time, because there are 4 squares in this Punnett square here, where she would end up with both recessive alleles. Thank you so much for watching, and these are my references.